Farmers are always seeking ways of maximising their crop production. This is mainly done by selectively breeding crops with favourable genetics and using pesticides or fertilisers. One problem that is incurred with the use of fertilisers and pesticides is insect resistance. In this case, more chemicals must be used and there is a higher negative environmental impact. Usually, genetic engineering is used to create higher yields, but in the 1980s, the first transgenic plant was made by inserting Bacillus thuringiensis or Bt bacteria, which produces a chemical that kills insects. Over the recent decades, the method has worked, but it has left many farmers and researchers wondering, what if the insects became resistant to Bt bacteria? It can be seen in some cotton plantations that insects have become somewhat resistant and therefore stronger Bt cotton crops were planted. This means a high toxicity level, which is bad for the environment. Another way of reducing the Bt resistance is by planting refuges alongside the Bt cotton crop. This refuge gets infested rather than the cotton crop section itself, and this method has proven to reduce the evolution of Bt resistance in bugs, such as the bollworm whose larvae can cause extensive damage. Although refuges do work, they cost a lot of money and they reduce the amount of crops viable for use. The aim of this study is to see what methods or method would be better and more effective economically and production-wise. The methods used in this report were A. The use of Bt cotton crops without a refuge, B. The release of sterile male moths on non-Bt cotton crops, C. The release of sterile male moths on Bt cotton crops. The report found that genetically engineered Bt cotton produces Bt toxin that kills moths, but many will develop a resistant allele to the toxin unless a refuge of non-Bt cotton is planted. Non-Bt cotton was protected by the release of sterile male moths that overwhelmed the healthy male moth population. Although there was a change from severe to minimal damage of the crops, the release of large numbers of sterile moths was found costly. The Bt cotton combined with the moderate sterile male numbers released was found cheaper, and it provided the most favourable crop protection with no emergence of moths containing the Bt resistant allele. Computer simulations were also used to determine what the effects of different release rates would have on large-scale field trials. The three sterile release rates were none, very, low, and low. Simulations showed that when sufficient numbers were released, the population of bollworms did not continue or develop a resistance over a 20-year period. It can be seen that the resistance evolved within 3 years without the release of sterile moths and refuges. Moreover, the populations did not grow or develop a resistant allele with the weekly low releases of sterile moths. The rate of resistance evolution was decreased with the number of sterile moths released, the refuge percentage of total crops, or a combination of both. The main finding in this report was that the release of moderate numbers of sterile moths plus refuges was more economically and environmentally viable. This article has been cited 40 times, and many other articles have similar methods or results. This article has an update, and it is called From the Integrated Pest Management to Integrated Pest Eradication Technologies and Future Needs by Suckling et al., which found that by using biological interventions such as sterile moths, there was an increase in crop yield and a decrease in economical and environmental impacts.